Carl Sagan wrote in Cosmos, "Life is tenacious beyond our imagination. Extremophiles are thriving in impossible places. Deep ocean, polar regions, radioactive zones. Life's limits are constantly expanding." This discovery is at the heart of Carl Sagan's astrobiology vision. Life can exist not only in moderate conditions, but also in the most extreme environments. Every new extremophile discovery increases the possibility of life in the galaxy. In the 1970s, Carl Sagan pioneered extremophile research. While designing Viking Mars missions, he emphasized the diversity of life. He was developing a life finds a way philosophy. "Life can exist in places we don't expect," Sagan said. "By studying Earth's extreme environments, we can gain understanding of alien worlds. Tardigrades, water bears, are the most resilient organism discovery. First observed by Anton van Leeuwenhoek in 1773, but extreme survival capabilities were recently understood. 1 millimeter long, 8-legged microscopic animal, bear-like appearance, hence the nickname water bears. But their superpowers are incredible. They can survive in the vacuum of space for 10 days. They tolerate absolute zero temperature, Miters 273 books. They withstand boiling water temperature. They survive pressure 6,000 x atmospheric levels. They tolerate radiation 1,000 times the lethal human dose. X-rays, gamma rays, ultraviolet exposure at extreme levels. DNA damage repair mechanisms are sophisticated. NASA conducted the TARDIS experiment on Space Shuttle Discovery in 2007. Tardigrades in Space Program. 12 days exposure on International Space Station external platform. Vacuum. Temperature extremes -157 Toll plus 121 cross. Solar radiation, cosmic rays. Direct exposure to harsh space environment. Analysis after return to Earth was shocking. 68% of tardigrades survived. 12% showed complete recovery. Reproduction capability was maintained. Some eggs laid in space conditions were viable. How is this possible? Tardigrades enter a cryptobiosis state. Greek meaning hidden life. Metabolic suspension, cessation of animation. Dehydration process, losing 85% of body water. Preserving cellular structure while metabolism stops. Tune state, minimum between life and death. Trehalose sugar accumulates in high concentration. Cellular protection from extreme dehydration. Glass-like state, preserving cellular components. DNA repair mechanisms are enhanced. Damage suppressor protein D-sup, unique to tardigrades. DNA break repair efficiency is incredible. They can remain in tune state for years. World record, 120-year-old museum specimen revived. Addition of water restarts metabolism. Carl Sagan described this resilience as life's most remarkable achievement. He wrote in Cosmos, "If life can survive in Earth's most extreme conditions, it's possible on other planets too." Deep ocean thermal vents are completely different extreme environments. First discovered by Alvin submersible at Galapagos Rift in 4,000 meters depth. Zero sunlight penetration. Eternal complete darkness. Pressure 400 times atmospheric level, crushing. Temperature 400 Ross, hydrothermal vent water. Sulfur, methane, hydrogen sulfide in toxic concentrations. Extreme heavy metal contamination. Highly acidic pH conditions. Minimal oxygen levels. Toxic chemical cocktail lethal to surface life. But thriving ecosystems were discovered in this hell. Giant tube worms 2 meters in length. Ghost white crabs scavenging. Bizarre fish species with unique adaptations. Chemosynthesis independence from sunlight. Bacteria converting chemical energy from hydrogen sulfide and methane. Primary producers as ecosystem foundation. Alvinella pompeiana, Pompeii worm, most heat-tolerant complex organism, thriving in 80 tertiary water temperature. Normal protein denaturation occurs at 60 tertiary, but specialized adaptation. Heat shock proteins, HSPs, prevent temperature damage. Chaperone molecules maintain protein folding. Modified cellular membrane composition. Symbiotic bacteria provide protective coating. Heat buffer insulation from external temperature extremes. Mutualistic relationship survival strategy. Pyrococcus furiosus hyperthermophile bacteria. Optimal growth at 100 Cers. Boiling water is preferred environment. Heat-stable variant of DNA gyrase enzyme. Thermostable enzymes have enormous industrial applications. PCR amplification, detergent production, food processing, enabling biotechnology revolution. Carl Sagan immediately recognized Europa Moon connection. Jupiter satellite with ice-covered subsurface ocean. Tidal heating makes geothermal activity probable. Europa thermal vents possible analog to Earth deep ocean, wrote Sagan. Life could thrive in similar extreme conditions. Galileo spacecraft 1990s Europa magnetic field data. Subsurface conducting layer indicating salty ocean. Ice shell thickness estimated 15 to 25 kilometers. Europa ocean volume 2 to 3x Earth's total ocean. Liquid water maintenance for 4.5 billion years.
Chemical energy sources probable. Radioactivity from Jupiter's magnetosphere. Tidal flexing internal heating. Seafloor hydrothermal activity likely. Earth thermal vent analog conditions. Antarctica extreme cold environment research advancing. Lake Vostok subglacial lake buried under 4 kilometers ice. Complete isolation for 15 million years.
Russian drilling project obtained water sample in 2012. Bacterial DNA sequences identified. Psychrophile cold-loving organisms living. These bacteria experienced 15 million years of evolutionary isolation. Development of unique genetic adaptations. Antifreeze proteins prevent ice crystal formation. Cold shock proteins maintain cellular function at low temperatures. Membrane fluidity preservation. Cold optimization of enzyme activity. Don Juan Pond, Antarctica's saltiest natural water body. 40% salt concentration. 10x seawater. Freeze prevention at minus 50 Vislaro temperature. Proof of microorganisms living in hypersaline environment. Holophile salt-loving extremophile. Extraordinary adaptation. Unique osmotic pressure regulation mechanism. Atacama Desert, Chile, ultimate dryness testing ground. 1 millimeter annual rainfall. Extreme aridity. Mars condition analog research. NASA Mars rover field testing location. Intense UV radiation exposure. Extreme temperature fluctuation. Soil chemistry. Mars similarity. Even here endolithic bacteria discovered. Habitat inside rock crystal matrix. UV protection from mineral shield. Minimal water requirement. These bacteria hide in crystal lattice. Photosynthesis in minimal light conditions. Extremely slow metabolism. Centuries between divisions. Quartz. Feldspar crystal structure colonization. Cryptoendolithic lifestyle. Avoiding surface. Microscopic ecosystem in rock interior. Deinococcus radiodurans, Conan bacterium, ultimate radiation resistance, survival of 5,000 gray G radiation dose, human lethal dose, 5G Ry comparison, 1000x radiation resistance mechanism unique, extraordinary efficiency DNA repair system, multiple chromosome copies provide DNA redundancy, advanced recombinational DNA repair pathway, radiprotein specialized function, incredible level of damage tolerance, Chernobyl disaster aftermath research site. 1986 nuclear accident, extreme radiation contamination, exclusion zone organism study, fascinating, discovery of melanin-containing fungi utilizing radioactivity, Cryptococcus neoformans, Wangella dermatitis species identification, radiosynthesis analog process to photosynthesis, melanin pigment absorbs gamma radiation, energy conversion for glucose production, plants utilize sunlight energy, these fungi use radiation as energy source, discovery of completely novel metabolic pathway, incredible diversity of life's energy sources, observation of melanin-rich fungi growth on reactor walls, correlation between radiation level increase and fungi growth rate, unique positive response to radioactivity, pH extremes chemical challenge for life, battery acid pH 0 highly corrosive, household bleach pH 14, caustic alkaline, most life requires pH 6-8 neutral range, protein denaturation, enzyme inactivation consequence of pH extremes, ferroplasma acid hermanus thriving in pH 0 environment, natural occurrence in acid mine drainage habitat, Iron Mountain, California research location, energy generation through sulfur oxidation and iron metabolism, extraordinary acid tolerance mechanisms, unique adaptation for cellular pH regulation, alkaline soda lake environment opposite extreme, pH 12 to 14 soap-like caustic conditions. Examples include Mono Lake, California, Lake Magadi, Kenya, Natronobacterium gregorii alkalophile bacteria thriving, high sodium carbonate concentration tolerance, specialized alkaline adaptation mechanisms, modified cellular membrane composition, buffering system for internal pH regulation, alkali tolerance optimization of enzymes, pressure extremes at Mariana Trench deepest ocean location, Challenger deep 11 kilometers depth, 1000x atmospheric pressure crushing force, Hodl zone life previously thought impossible, recent research discovered thriving ecosystems, abundant snailfish, amphipods, microorganisms, piezophile pressure-loving organism, unique adaptations, protein structure modification for pressure resistance, cellular membrane pressure tolerance, barophilic bacteria in deep sediment habitats, pressure requirement for optimal growth, surface condition intolerance due to decompression sensitivity, temperature extremes in both directions challenging life, amazing preservation of ancient bacteria in permafrost, incredible survival from Pleistocene epoch, revival of 400,000-year-old bacteria from Siberian permafrost,
Carnobacterium pleistocenium species identification, natural freezing cryopreservation demonstration, bacterial spore viability in preserved mammoth carcasses, activation through thaw process, metabolic restart, record-breaking longevity survival, antifreeze proteins and cell membranes prevent ice crystals, cryoprotectant molecules minimize cellular damage, sophisticated cold adaptation mechanisms, high temperature extremes equally remarkable, strain 121 bacteria growth temperature record, survival at 121 6-degree autoclave sterilization temperature, incredible adaptation of hyperthermophile heat-loving organisms, deep-sea hydrothermal vent habitat, extreme level of protein thermostability, Methanopurus conluri current temperature record holder, demonstration of optimal growth at 122 6-degree, unique heat tolerance mechanisms,
protein stabilization through disulfide bonds, thermal optimization of amino acid composition, enhancement of cellular component heat resistance. Carl Sagan recognized revolutionary impact of discoveries for exobiology. He wrote in pale blue dot, "Every extremophile discovery increases the possibility of alien life. Astrobiology field providing extremophile research foundation, expansion of habitability definition, challenge to Goldilocks zone concept, traditional habitability narrow temperature range, surface liquid water. Extremophile research reveals subsurface habitat, alternative biochemistry possibilities. Mars exploration guided by extremophile research methodology, subsurface life detection strategy, Earth analog environment study. Carl Sagan's significant involvement in Viking missions, life detection experiment design incorporating extremophile knowledge, labeled release experiment controversial results, organic compound detection, debate over biological versus chemical origin. Carl Sagan's application of extraordinary claims require extraordinary evidence principle, cautious approach to ambiguous result interpretation, modern Mars research, benefiting from extremophile discovery context, Curiosity rover organic molecule detection, observation of seasonal methane variation, probable subsurface habitat with radiation protection, permafrost ice as water resource, geothermal heating possibility, Perseverance rover sample collection planned for future Earth return analysis, extremophile analog comparison planned, Europa Clipper mission 2024 launch ice penetrating radar subsurface ocean mapping, extremophile Earth analog informing mission design, plume sampling for organic chemistry analysis, possibility of amino acid complex molecule detection, Europa ocean volume estimated 2 to 3x Earth's ocean, probable chemical energy sources from hydrothermal activity, Enceladus geysers organic molecule discovery from Tiger Stripe region, Cassini spacecraft amino acid detection, evidence of complex chemistry in subsurface ocean, salinity, temperature, pressure condition extremophile analog comparison, assessment of life survival possibility, Titan methane lakes, alternative biochemistry laboratory, carbon chemistry methane solvent system, completely different analog to water-based life, extremophile research illuminating alternative pathway possibilities, theoretical framework for silicon-based, sulfur-based, methane-based biochemistry, TRAPPIST-1 system discovery of 7 Earth-sized planets, multiple locations in potentially habitable zone, challenges of tidal locking and radiation exposure, application of extremophile survival strategies, tolerance for day-night temperature extremes, James Webb Telescope atmospheric biosignature detection capabilities, traditional signatures like oxygen, water vapor, methane combinations, possibility of extremophile metabolism producing alternative biosignatures, sulfur compounds, methane production, unusual atmospheric compositions, Carl Sagan's vision for incorporating extremophile insights into SETI approach, traditional radio signal search methods, possibility of alternative lifeform communication methods, chemical signals, radiation patterns, unusual electromagnetic emissions, breakthrough listen project, comprehensive search considering extremophiles, possibility of silicon-based intelligence, radiation-powered organism communication, laboratory extremophile cultivation technique advancement, improved bioreactor extreme condition simulation, creation of controlled environments for temperature, pressure, pH, radiation, salinity, research methodology standardization, industrial applications in enzyme production, enormous biotechnology revolution applications for extreme temperature, pH tolerance, applications in PCR amplification, pharmaceutical production, environmental remediation, astrobiology field laboratory extreme environment simulation, Devon Island Arctic Mars Analog Research Station, Atacama Desert Mars Mission Training Simulation, Antarctica Ice Drilling Subsurface Exploration Technology, genetic sequencing revelation of extremophile adaptation mechanisms, identification of heat shock proteins, antifreeze compounds, radiation resistance genes, horizontal gene transfer sharing extremophile adaptations, bacterial conjugation transformation transduction mechanisms, synthetic biology extremophile engineering app,